All right, we're picking up with Porifera. Porifera is the phylum name for the sponge. Sponges are aquatic organisms. I think they're pretty much all marine, meaning they live in the ocean. And they're probably one of the simplest animals there are. The sponge itself is notable because it has no symmetry and no true tissues. So it's essentially a giant collection of cells that are sort of hanging out together. Sponges are immobile, meaning they cannot move. They root to a structure and they stay there. And you can think of a sponge sort of like a vase with holes in the sides, right? So water flows through the holes in the sides into the main cavity of the vase. And then it flows out through the top. And that is how water moves through a sponge. And the idea is that water moving through the pores into the central cavity brings with it nutrients. And then waste can be excreted into the water in the cavity and expelled out the top. So that basically you have a constant flow of water bringing nutrients into the sponge and taking waste out of the sponge. And this helps keep the sponge alive despite the fact that it cannot move and search out food. It brings the food to it. You can think of a sponge as kind of a colony of cells working together. So there are two basic types of cells I want you to know when you think sponge, and one is the choanocyte. The choanocyte is a flagellar cell, meaning that it has a flagella. A flagella is a little tail-like appendage on the cell. So if you remember way back to cell organelles, the cytoplasmic extension, the flagella sort of spins around like this, right? And it collects food. So the flagellar cells, the choanocytes, spin their flagella, and that allows water to move. So the choanocytes are on the inner wall of the sponge. They're facing the inside of it, and they're spinning their flagella around. And that's pulling water in through the pores. And at the same time, those flagella gather the nutrients in the water into the choanocyte. And so the choanocyte is able to absorb the nutrients. And then the choanocyte uses what nutrients it needs and then passes the rest of the nutrients on to the amoebocyte. The amoebocyte is sort of a free-moving cell. It moves around within the sponge. And basically, it is the food distribution cell. It moves all through the sponge, delivering food to the different cells. And that's how it keeps all the sponge alive. In addition to collecting, digesting food, and moving nutrients between the cells in the sponge, the amoebocytes also serve an important function in that they produce the skeletal fibers. Got the air quotes going on there because it's not skeletal like when we think of a skeletal, we think of a bone. Rather more like it's a little mineral that's distributed all throughout the sponge. And these little minerals basically help provide rigidity, structure to the sponge. They reinforce it. So here's a picture of your average sponge here. And you can see I've got a cutaway there showing the inside of the vase, right? And so we've got these pores that water flows into the sponge. And then this right here is a choanocyte. And this is its flagella. And it spins its flagellum and pulls water and nutrients over to the cell. It absorbs the nutrients and then passes them to the amoebocyte, which travels up and down the sponge. And then I also have these little pictures pointed out here for spicules. Those are those skeletal fibers, but you don't have to remember the word spicule. So here are a large variety of different sponges. These are all different species of sponge that you can see, and they're all very interesting organisms. This one here looks much less like a vase, but it still has a central cavity through which water will flow and then be expelled. So this particular type of sponge when fished out of the water and dried out, will end up looking like the picture down here in the lower left. And that picture in the lower left there is of a natural sponge. In fact, the sponge that you use to clean off your dishes is synthetic. It's not natural. But if you were alive 50 years ago, you would be using this, the natural dried up sea sponge. All the original sponges people used in cleaning their dishes and their house and whatnot, these were all natural sea sponges. And so that's sort of what's left behind when you let all the cells die off. That's a whole lot of these little skeletal fibers, these spicules right there. It can hold a lot of water. Anyway, so now that we're going to move on past sponges, it's time to talk about digestive openings. So everything past the sponge is going to have a simple digestive tract. Well, for the most part. The phylum themselves. Individual species might have interesting alterations, but every phylum past the sponge will have a digestive tract. And one of the ways we differentiate amongst the types of organisms, the types of phyla, is the number of openings in their digestive system. So they might have one opening, which serves as both the mouth and the anus, and it opens into the gastrovascular cavity. Basically, the gastrovascular cavity is a hollow space. So the digestive tract is the opening into the gastrovascular cavity. And that hollow space is where digestion occurs. At the same time, waste leaves that cavity and gametes leave that cavity and exit through our single opening. The gastrovascular cavity has that opening, and that's the universal pore. Food goes in, waste and gametes come out. Our alternative to the single opening is the two openings. A digestive tract with two openings has a mouth and an anus. And so it's a complete tube running the length of the organism. So remember when the blastula, I said there's two involutions? Well, so when I was talking about the blastula, I talked about how it was a hollow ball of cells, right? Big ball of cells. And then you had an involution, a pore opening up into it, like poking a balloon, right? And I said there were two involutions. Well, there's one involution for an organism that has a single gastrovascular cavity. Only one involution. Two involutions in the gastrula occur when you are having a complete digestive tract. So we have a separate mouth and anus. So we might call a single opening an incomplete digestive tract and two openings a complete digestive tract. All right. So now that we've talked about openings, let's look at Cnidaria. They have radial symmetry, pizza slices. They have true tissues. All right. First phylum where we see true tissues. They have an ectoderm, which is sort of the outer covering, the skin, and they have an endoderm, which makes up the digestive cavity, which is a gastrovascular cavity, meaning Cnidaria have one digestive opening. They have an incomplete digestive system. Food goes in, waste and gametes come out. They are bell-shaped with tentacles around the opening of the gastrovascular cavity. So there are two basic types of cnidarians. Oh, getting ahead of myself. So all those tentacles have little stinging cells. And we call these stinging cells cnidocytes. Cyte meaning cell, cnido, cnidaria. So these cnidocytes are stinging cells, and they essentially look like little harpoons. They have that little pointy end with a barb to make it so that once it shoots into something, it doesn't easily come back out. And basically, any kind of pressure at all on the tentacle will cause all of these stinging cells to fire their harpoons out. And so they'll penetrate the skin of whatever they're in contact with. And they can poison any prey. They deliver a toxin. So Cnidarians can be pretty nasty. They are all venomous. They are poisonous. A lot of them aren't extremely dangerous to us. Most sea anemones, we can touch them without any kind of discomfort. Our skin is thick enough that their harpoons can't shoot all the way through it. Some jellyfish, not so much. Jellyfish can deliver a pretty nasty sting. Okay, so there are two basic body plans to phylum Cnidaria. One is the polyp. The polyp is immobile. It's rooted to the ground. And think of it like an upside-down bell. The mouth is up and the tentacles face up like this, right? If you've ever seen a sea anemone, this is the polyp. Sea anemones are polyps. Then there's the medusa. The medusa is the bell in its correct shape with the tentacles facing down and the mouth facing down. And that's your jellyfish. Jellyfish are the medusa. So, polyps are adult sea anemones. Medusas are jellyfish. What's interesting about jellyfish is that after hatching, they go through a polyp stage. So the first stage of life for jellyfish is an immobile polyp stage, which is kind of neat. And a lot of sea anemones have a first stage of life after hatching that is a medusa stage before settling down. So that's kind of neat. So here's a variety of organisms. I have sea anemones on top, and then I have jellyfish on bottom. So you can see the medusas on the bottom, the polyps on the top. All right, now that we've talked about jellyfish, it's time to talk about a new feature in differentiating amongst the phylum. So now we're going to talk about the body cavity. A body cavity is a cavity or a hollow space inside an organism. You can have a body cavity that is partially to fully lined by mesoderm. So mesoderm is that middle tissue. So it may only be partially lined by mesoderm, or it may be fully lined by mesoderm. And basically it separates the outer layers of the organism from the organs. So if you think about humans, we have that hollow space in our belly and under our rib cage. That is a body cavity. And that big hollow space in there contains all our organs. So we have our outside, then we have that hollow space, and then we have all the organs in there. So the digestive tract and the organs are inside the body cavity. There are three different types there are three different terms to describe whether or not an organism has a body cavity and what kind of body cavity it has. The first term is acoelomate. Acoelomate organisms have no coelom. The coelom is the body cavity, by the way. So I thought I might throw that out there since I'm just suddenly saying coelom. So coelom is body cavity. Acoelomates have no body cavity. Pseudocoelomates have a body cavity that is not completely lined by mesoderm. What does that mean? That means there's your external tissues and then mesoderm, and then there's all the organs in there. Whereas the coelomate organisms have fully lined by mesoderm. What does that mean? There's your external tissues, then there's the mesoderm, and then the hollow cavity, and then all the organs have mesoderm around them. That's probably 10 minutes. So we'll pick up with Mollusca. and talking about what kind of body cavity they have. Waiting with bated breath you are, I know. Got some Yoda talk in there. That was weird.